Disclaimer: this podcast is for educational purposes only and is not medical advice. Always consult with a qualified healthcare professional before starting or changing any treatment. Today we are going to spend some time diving deeply into dermorphin peptides. Dermorphin is one of those fascinating natural compounds that draws attention not only because of its unique origins, but also because of the questions it raises about pain, biology, and medicine. To understand dermorphin, we have to start with its discovery. In the 1980s, researchers studying frogs from South America, specifically from the Amazon basin, noticed that their skin secretions contained a wide variety of bioactive peptides. Among them was dermorphin, a peptide that turned out to have very strong activity on the opioid receptors in mammals. The name itself, dermorphin, comes from derma for skin and morphin to reflect its similarity to morphine in function. What makes dermorphin so unusual is its potency. Laboratory studies revealed that it binds to mu-opioid receptors much more strongly than morphine does. In fact, it has been reported to be many times more powerful in terms of analgesic effect. This discovery was both exciting and concerning. On one hand, the idea that nature produced a compound more potent than morphine suggested new therapeutic possibilities for managing pain. On the other hand, such potency raised red flags about safety, misuse, and the potential for dependence. Dermorphin is a peptide consisting of 7 amino acids, what scientists call a heptapeptide. This short chain structure is arranged in such a way that it has a high affinity for the mu-opioid receptor. The exact sequence is unusual and contributes to its powerful activity. Because peptides are made of amino acids, they often degrade quickly in the body, but dermorphin has structural characteristics that allow it to resist breakdown and to cross biological barriers more effectively than many other peptides. This explains in part why it can be so active even at very low doses. When we talk about dermorphin, it is also important to place it in the larger context of opioid biology. Our brains and bodies have natural systems designed to regulate pain, stress, and reward. Endorphins are the natural opioids that our bodies produce internally. Externally, substances like morphine, heroin, fentanyl, and in this case dermorphin, can also bind to those same receptors. The difference lies in potency, duration, and side effect profile. Dermorphin is interesting because it shows how evolution in a completely different species produced a peptide that interfaces with human receptors in a profound way. One of the questions researchers have asked is whether dermorphin could one day be developed into a therapeutic drug. Imagine if you could treat severe pain with a smaller dose and perhaps with fewer side effects. However, the reality is more complicated. Potency does not always translate into safety. With dermorphin, studies showed it is indeed highly effective in animals, but questions about respiratory depression, tolerance, and dependence remain unanswered in humans. Because of that, dermorphin has not been approved for medical use. An interesting, though controversial, episode in dermorphin's story came from horse racing. Reports emerged that dermorphin was being used as a doping agent in racehorses. The reasoning was that by reducing pain sensitivity, the animals could perform harder without stopping. This is not only unethical, but also dangerous. Pain is a protective mechanism, and removing that signal without addressing underlying issues can lead to harm. As a result, Testing and bans were implemented in racing circuits to prevent dermorphin misuse. Despite these controversies, dermorphin remains a subject of ongoing research. Its extreme potency makes it a useful tool for scientists who want to study the opioid system more closely. By comparing how dermorphin interacts with receptors to how morphine or endorphins interact, researchers can learn more about receptor subtypes, binding affinities, and downstream signaling pathways. In this sense, dermorphin is not just a potential drug candidate, but also a scientific probe that helps map out the complexities of the opioid system. Another layer of interest lies in the origin of dermorphin. Why would a frog produce such a compound in its skin secretions? The answer likely has to do with defense. Frogs in the Amazon basin face a wide range of predators and microbial threats. By producing peptides that have strong biological effects, they increase their chances of survival. Some peptides defend against bacteria and fungi, while others, like dermorphin, may discourage predators through toxic or disorienting effects. This illustrates the incredible diversity of natural chemical defenses and the potential treasure trove of compounds waiting to be studied. From a biochemical standpoint, dermorphin shows how a small change in peptide sequence can have a massive impact on biological activity. Even though it is only 7 amino acids long, the arrangement of those amino acids makes all the difference. This is why peptide research is such a growing field. By modifying natural sequences or designing synthetic versions, scientists hope to create new molecules that combine the best features of natural compounds with improved safety and stability. As we continue, let's think about the larger lessons from dermorphin. First, it shows the power of natural products. Time and again, compounds from plants, animals, and microbes have provided leads for some of the most important medicines in history, from antibiotics to anti-cancer agents. Dermorphin adds to that list as an example of how even a small peptide from frog skin can open doors to new research. Second, dermorphin highlights the double-edged sword of potency. Stronger is not always better. In medicine, balance is crucial. A compound may be powerful, but if it carries too high a risk, it may never be suitable as a therapy. This is why researchers must carefully study not only the benefits but also the dangers of every new discovery. Third, dermorphin connects to the broader conversation about the opioid crisis. In many parts of the world, opioid misuse has become a public health emergency. Against that backdrop, the discovery of an even more potent opioid like dermorphin can seem alarming. However, it also reminds us of the importance of continued research. By understanding these compounds, we may eventually design better, safer alternatives that relieve pain without the same risks. To fill out our understanding, let's talk about what research has revealed in animal models. In experiments, dermorphin produced profound analgesia, meaning pain relief, at doses far lower than morphine. Its effects were long-lasting, which is another point of difference. But it also caused typical opioid side effects such as sedation and reduced motor activity. These findings suggested both promise and limitation. Further testing in primates confirmed high potency, but also underscored the risks associated with such strong receptor activation. One area of speculation is whether dermorphin or related peptides could be modified to separate pain relief from the addictive or sedative effects. This is sometimes called the search for biased agonists, molecules that trigger only certain signaling pathways downstream of the receptor. If researchers could harness dermorphin's analgesic strength while avoiding pathways that cause dependence or respiratory depression, it could inspire a new class of painkillers. This remains a goal for the future. Beyond medicine, dermorphin also fascinates evolutionary biologists. The fact that a frog evolved a peptide with such specific and powerful effects on mammalian receptors is a reminder of the interconnectedness of life. Proteins and receptors often share evolutionary origins, and sometimes a compound in one species can unexpectedly influence another. Dermorphin is a striking example of that cross-species connection. Now, let's consider the ethical dimensions. Whenever we discover a powerful natural compound, questions arise about conservation and respect for the source. The frogs that produce dermorphin are part of delicate ecosystems in the Amazon. Protecting those environments ensures not only the survival of the species, but also the potential for future discoveries. Bioprospecting, or searching for useful compounds in nature, must be balanced with sustainability and respect for indigenous knowledge and biodiversity. As we reflect on dermorphin, it is worth recognizing how much remains unknown. Because it has not been developed into a drug, there are limits to human research. Much of what we understand comes from animal studies, biochemical assays, and receptor modeling. The gap between laboratory discovery and clinical application is wide, and dermorphin is still on the discovery side of that divide. Nevertheless, the story of dermorphin continues to spark curiosity. Whether it ends up as a drug, a research tool, or simply a fascinating chapter in peptide science, it teaches us about the richness of natural chemistry and the importance of studying even the most unlikely sources. The skin of a frog in the Amazon might seem far removed from human medicine, but as dermorphin shows, nature's molecules can reach across that distance and challenge our understanding. As we wrap up, remember that dermorphin is one of many peptides being studied today. Each has its own story, its own potential, and its own risks. By learning about them, we not only expand our scientific knowledge but also gain insight into how our bodies work and how nature has solved similar problems in different ways. Disclaimer: this podcast is for educational purposes only and is not medical advice. Always consult with a qualified healthcare professional before starting or changing any treatment. Thanks for listening, keep learning, and stay curious.